Hello, my name is Patryk Dabek. Today I'm going to be presenting my biology research project to you, but just to give a little bit of background about myself, I'm a senior at the Bergen County Academies in Hackensack, where I study science and technology. In my free time, I like to volunteer as an EMT in my hometown, and I'm planning to go to college next year, majoring in public health and molecular biology. To let you know a little bit about where my project stems from, it was during my freshman year biology class, I became curious about the metabolism of cells, and my research into cancer metabolism stemmed from there. My current project focuses on exploiting metabolic reprogramming in liver cancer and identifying the mitochondrial membrane potential response to selective ATP hydrolysis inhibition. So just to give a little bit of background information, my study deals with hepatocellular carcinoma, which is the most common type of liver cancer and is the third most common cause of cancer-related death worldwide. One of the biggest complications in treating hepatocellular carcinoma are the underlying malignancies of the liver. The overwhelming majority of patients have some type of underlying cirrhosis of the liver, which can be caused by a variety of things, including viral hepatitis, cystic fibrosis, hemochromatosis, and many more conditions. In turn, these conditions make hepatocellular carcinoma so much more difficult to treat. This is because traditional therapies struggle to maintain post-treatment liver function. One of the hallmarks of cancer is metabolic reprogramming. In the presence of oxygen, normal cells would use oxidative phosphorylation to generate the maximum number of ATP. However, cancer cells use glycolysis, meaning that they generate less ATP. However, this serves a purpose. By utilizing glycolysis, they can shuttle metabolic intermediaries into the macromolecular biosynthesis pathways that allow cancer to proliferate so rapidly. Like all cells, cancer cells need to maintain their mitochondrial membrane potential to prevent cell death. Since the electron transport chain is no longer pumping protons into the intermembrane space, cancer cells need to find a different way to maintain this gradient. They are forced to use the reverse action of ATP synthase, ATP hydrolase, to maintain this gradient potential. Since ATP synthase is a dual action rotor, one direction of the enzyme can be selectively inhibited by having a small molecule bind to the rotor in a specific way. I first learned about this dual action mechanism when I was doing research for a presentation about heart attacks. I was trying to figure out why the nutrients in the heart cells were being used up so rapidly. After a bit of digging, I found a journal article that showed that under ischemic conditions, myocardial cells switch the direction of their ATP synthase, thus hydrolyzing ATP. After some more research, I found that there was a pharmaceutical company working on developing a drug that would selectively inhibit ATP hydrolysis for the purpose of stopping the depletion of nutrients in the myocardial cells. As previously mentioned, since cancer cells are reliant on ATP hydrolase to maintain their mitochondrial membrane potential and stay alive, I decided that this drug could be applicable as a cancer treatment. Therefore, I wanted to study the effects of ATP hydrolase inhibition on cellular metabolism, mitochondrial membrane potential, and cellular proliferation. I hypothesized that if ATP hydrolysis was inhibited, then the mitochondrial membrane potential would in turn collapse, releasing pro-apoptotic proteins into the cytosol, inducing apoptosis, and targeting the growth-supportive microenvironment of cancer tumors. Fundamentally, this study sought to demonstrate and investigate the ability of selective inhibition of ATP hydrolase to target mitochondrial membrane potential and decrease cell proliferation. Feel free to pause on this slide that goes more into depth on the procedures that I used, but now I'm going to be talking about the results that I had. To examine the metabolic changes that occurred in cancer cells after treatment, I looked at glucose uptake and differences in ATP levels. These both decreased, showcasing that cells became less metabolically active and that ATP was being hydrolyzed inside of the cells. Furthermore, I conducted a flow cytometry experiment to look at the effect of treatment on cell cycle, and it was found that selective ATP hydrolysis inhibition actually induced a GOG1 cell cycle arrest, even though the other controls did not, meaning that this treatment prohibits cells from proliferating rapidly. To analyze the effect of treatment on cellular proliferation and viability, I conducted a MTS assay which uses a tetrazolium salt to quantitatively deduce if cellular mitochondria are active. Over 24, 48, and 72 hours, selective ATP hydrolase inhibition did in fact lead to a decrease in mitochondrial activity. Additionally, cell death increased based on a trypan blue exclusion assay and an increase in reactive oxygen species production. All this data tells us that in fact the cancer cells are entering apoptosis, but now we need to figure out why. The mitochondrial permeability transition pore works by opening the voltage-dependent anion channel when the mitochondrial membrane potential collapses. This induces the release of cytochrome C, which recruits APAF1 and procaspase 9, forming the apoptosome and leading to the activation of the executioner caspases 3 and 7, thus inducing apoptosis. To qualitatively determine mitochondrial permeability transition pore opening, we need to look at cytochrome C levels in the mitochondria and the cytosol. Through the use of subcellular fractionations, we can determine the kinetics of cytochrome C. Looking at the graph on the left, we can see that as soon as cells are treated, cytochrome C levels increase in the cytosol. This corresponds to an increase in the executioner caspase levels, meaning that in fact the depolarization of mitochondrial membrane potential is leading to the activation of apoptosis. To analyze the kinetics of mitochondrial membrane potential in the cell, I utilized CMX-rosamine, a dye which fluorescence is based off of accumulation, which increases with higher hyperpolarization. The findings of this experiment show that BMS 199264 significantly collapses mitochondrial membrane potential in cancer cells. To analyze these pictures, I used Fiji and ImageJ software to isolate the red color channel and measure the fluorescence of CMX-rosamine in the cells. Furthermore, statistical data analysis was performed on all of my assays to determine if they were statistically significant by using a t-test in Excel. I've also attached annotated diagrams throughout this presentation to help better describe my research. The findings of this study have many applications. Exploiting metabolism, this treatment creates the possibility for a whole new class of drugs that target biosynthesis pathways in cancer. Furthermore, selectively utilizing a pathway found only in cancer cells creates a very personalized treatment that can be used directly to treat liver cancer, decreasing the adverse reactions patients experience through non-specific medications, thus improving their quality of life. Furthermore, the findings of the study have great potential to be used in many different ways, from being linked to pharmaceutical moieties, targeting the hyperpolarized mitochondrial membrane of cancer cells. Lipophilic cations can attach to this drug and make it even more effective, creating endless possibilities for patient care. I'd like to give a big thanks to everyone in the Bergen County Academies and the Nanostructure Imaging Lab who have helped me with my research. This concludes my presentation. I hope you've enjoyed and you learned something new. Thank you for your time, and please enjoy the rest of your day. Thank you.